In a world where pharmaceutical production was often a complex and expensive endeavor, a groundbreaking innovation emerged: Prodigene corn. This remarkable corn variety had been genetically engineered to produce pharmaceutical proteins within its very own seeds, turning the humble crop into a potent biofactory. In the realm of medicine, there were times when large-scale production of pharmaceutical proteins became a pressing necessity. Vaccines, therapeutic proteins, antibodies, and enzymes were in high demand. The traditional methods of production, involving bioreactors and fermentation, were laborious, costly, and required meticulous control over the manufacturing environment. That's where Prodigene corn stepped in, offering a glimmer of hope and a new way forward. Prodigene corn held numerous advantages over conventional production methods. First and foremost, it boasted scalability. Corn, a staple crop grown abundantly across vast expanses of land, could now be harnessed to produce substantial quantities of pharmaceutical proteins. The sheer potential of this mass production made it an enticing option for pharmaceutical companies and researchers alike. Moreover, this innovative corn variety offered a cost-effective solution. The reliance on plants as biofactories eliminated the need for expensive infrastructure and specialized equipment. As a result, production costs could be significantly reduced, making pharmaceutical proteins more accessible to those in need. Safety was another paramount consideration. Prodigene corn operated within a contained system, ensuring that the pharmaceutical proteins were exclusively produced within the seeds themselves. This containment greatly minimized the risk of contamination or accidental release into the environment, providing reassurance to both scientists and the public. The journey to create Prodigene corn was not without its challenges. It began with the identification of the target protein, carefully selecting the specific pharmaceutical protein that held the promise of healing and saving lives. Through the marvels of genetic engineering, the gene responsible for producing this protein was isolated and inserted into the corn plant's genetic blueprint. The process was meticulous and precise. Gene cloning and transformation techniques were employed, delicately merging the desired genetic material with the corn plant's genome. The modified corn plants were then grown under controlled laboratory conditions, using tissue culture to regenerate entire plants from the modified cells. But it didn't stop there. The newly developed transgenic corn lines had to endure rigorous field trials. These trials evaluated various factors, such as protein expression levels, seed yield, and overall plant health. Only after successfully passing these tests would the chosen transgenic corn lines advance to the grand stage of commercial production. In vast fields stretching as far as the eye could see, Prodigene corn stood tall and proud. Carefully cultivated, monitored, and nurtured, these extraordinary corn plants blossomed into a pharmaceutical production powerhouse. They diligently produced the desired proteins within their seeds, which were then harvested and processed to extract the valuable pharmaceutical compounds. The impact of Prodigene corn was profound. It ushered in a new era of accessibility, breaking down the barriers that limited the availability of vital medicines. Its cost-effectiveness transformed the landscape of pharmaceutical production, allowing life-saving treatments to reach those who needed them most. The innovation embodied hope, resilience, and the relentless pursuit of scientific progress. In the annals of biotechnology, the development of Prodigene corn would forever be hailed as a monumental achievement. It symbolized the triumph of human ingenuity over challenges, offering a glimpse into a future where plants could be enlisted to play a vital role in the production of pharmaceutical miracles.